This lecture covers structure, replication, and expression of genes. But before I do that, I'll get into some background on the three major macromolecules and some essential terminology, and then a little bit of history before I jump into actual DNA replication, and then eventually transcription, and then eventually translation. So the three major macromolecules: DNA, which is the storage molecule for genetic instructions to carry out metabolism and reproduction, etc. All the code is stored up in the DNA. And as you'll see from some of the historical evidence, this was not always intuitive. It took a fair amount of important experimentation in order to deduce that DNA is the genetic material and not proteins, as probably a lot of people favored back at the time, because the number of amino acids, for example, far outnumber the number of nucleotides. And so it seems like you could have a more robust language with proteins than you could with DNA. RNA obviously expresses the information in DNA. So a piece of DNA is transcribed into messenger RNA as the informational molecule, and then the messenger RNA is translated into protein. Some of the terminology that goes along with this section is starts with genomes, which are basically all the DNA present in a cell or a virus. Bacteria and archaea generally have one set of genes and are haploid, with the exception of vibrios. And I wouldn't exactly call vibrios diploid, but they do have two copies of their chromosomes, but they're not exact copies, and so they're not true diploids. And as I'll show you, the term genotype is just a specific set of genes that an organism possesses, and we have specialized notation to describe the genotype of microbes that we make in the lab, for example, which I'll show you as well. And then phenotype is just the observable characteristics of a particular genotype. So the genotypes of some strains that I've worked on, for example, and my colleagues are listed here. You see ES114 is the wild type, so-called strain that comes from nature. And then you see another strain, KV7913, for example, that was constructed in the lab and has the genotype indicated here, where this particular gene encoding a nuclease is insertionally inactivated by a transposon that was obviously inserted into it, and therefore you have this particular genotype. The resulting phenotype then of this organism can be displayed on a differential medium like DNA agar. And so here's the wild type strain showing a halo where it's able to digest the DNA in this medium, whereas the nuclease mutant, specifically the XDS nuclease mutant, is unable and therefore does not have a halo around that colony. As far as cholera genome organization that I'd mentioned, this concept, normally microbes have a single large chromosome, usually circular, but not all. Some are linear, like the Borrelia, for example. And typically all the housekeeping functions are here on chromosome 1, and then primarily chromosome 2 is a smaller chromosome that often contains a lot of niche expanding characteristics. But another important feature that I wanted to point out here is that some of these things in red, these horizontally acquired novel pieces of genetic information that expand the genotypes of microbes that don't rely on, obviously, natural evolution, build up of point mutations and so forth. So, for example, cholera has a integrated phage. Not all strains, but toxigenic strains of cholera have an integrated phage called CTX-V. And on this chunk of DNA that was inserted by a virus infection includes, for example, cholera toxin that gives Vibrio cholerae its major virulence factor. Another horizontally acquired piece of DNA are pathogenicity islands. Here's a Vibrio pathogenicity island, specifically a set of genes that expands cholera's virulence. And then over here is a so-called integron island. These are often referred to as gene traps, and I'll show you that in a little bit more detail. So here's a representation of a integron. This leftward gene here expressed off of this promoter, the integron promoter, this leftward promoter, it produces an enzyme called an integrase. And so this enzyme can grab circular DNA cassettes with the ATTC insertion point here, can grab these things and insert them downstream of the insertion point that you see here. And so that gives this microbe a new gene, a potentially novel and important gene that can be expressed off of this rightward promoter that's actually embedded in the integrase. And then, as you can see here, additional genes can be grabbed by the integrase. And so you can essentially create these operons, if you will. And then if you notice, some of these genes can reassort, and basically the integrase can pull this out and put it in a different order. And obviously there's heavy selective pressure on these genes. If the order is not great, then it obviously will be selected against. If a particular gene is not useful, it will not favor reintegration in subsequent lineages. Here you see a Salmonella pathogenicity island, just a big hunk of DNA that encodes a whole bunch of different features. One of the main ones on this particular SPI1 is the type 3 secretion system apparatus that gives the Salmonella the ability to invade host cells. But you can see that there are other things on here as well, but these are massive hunks of DNA with lots of different genes. And again, this operon for the construction of a type 3 secretion system is an example of a pathogenicity island. So this was in Salmonella, and as I showed you in this example, there's also versions of this that are embedded in the Vibrio chromosomes as well. So here's an example of SPI1 in action. SPI1, those genes that I showed you here collectively produce the proteins necessary to make this needle, this type 3 secretion system apparatus, and the effector molecules as well. As if you look, you can see that that there are these effector molecules, the so-called CYPs, for example, that are also encoded on this pathogenicity island. So the needle is constructed, and then the pathogenicity island of effector molecules are inserted, which allows the microbe to reshape the apical surface of this particular cell and allow it to be taken into a vacuole. Notice that the bacteria also have a second pathogenicity island that they acquire that gives it another type 3 secretion system that produces additional effector molecules that then allow it to survive in this vacuole and not be killed. So again, the acquisition of these pathogenicity islands can be huge for expanding the niche that these microbes can occupy. So we can detect these foreign DNA insertions through a variety of means, and so basically this paraphrases what I've already said, that pathogenicity islands are often associated with transmissible DNA, and their presence correlates with pathogenicity, like the SPI1 and 2 that I just showed you. And a single PAI can transform a benign microbe into a virulent microbe in one massive fell swoop, as opposed to, again, waiting for millions of years of evolution to kick in. A microbe can acquire one of these and transform literally in minutes. But in order to identify these things, we can look at different features of them. For example, are they inserted into the 3' end of a tRNA that suggests that the hunk of DNA came from a phage? The term for that is transduction. Anytime novel DNA is acquired that modifies the genotype of a bacterium and it arises from a phage infection, we call that transduction. But sometimes, horizontal transmission can occur through conjugation or transformation. Conjugation is when the bacteria mate through a mating pilus, and transformation is when the bacteria just pick up free DNA from the environment. But they do have to have specialized machinery to bring that DNA in, and not all microbes can do that naturally. So this is, showing you some evidence how we would detect horizontal transfer. One thing we'd look for is differential GNC content of the piece of DNA that we think is foreign. And so again, usually the GNC content of the genome is fairly continuous or homogeneous, and so the microbe will often have a moderate G and C content because it's harder, obviously, to have these G triple bond C nucleotides. Usually you'd want something like that if you're trying to live in a high heat environment or something where you don't want your DNA to be denatured as easily. But generally, there's a fairly consistent ratio of these things throughout the genome. But then if you run into a patch of DNA where, the GNC content is significantly higher, you can surmise that it's probably came there relatively recently and hadn't had a chance to quite smooth out and have the same GC content as the rest of the genome. The other thing we can look for is codon usage. Obviously, the genetic code is degenerate, and there's multiple codons for single amino acids, and the microbes will have their preferred codons. But if you find a stretch of DNA that has unusual codon usage, odds are it came from someplace else, and again, the microbe hasn't had a chance to smooth that out. Are they flanked by repeats? These inserts, repeats often suggest sites of recombination. And then obviously the presence of phage genes, if there's capsid, protein, encoding genes, and so forth around the insert, odds are it came from a phage. Here I'm just showing you the different the fact that DNA is often supercoiled, and we typically will draw it as a simple circle like this, but in reality, there's different levels of supercoiling in bacterial genomes. Again, there are microbes that have a linear genome. And then an experiment like this just kind of illustrates that point. Here, you're just seeing different DNAs run on a agarose gel. And so here's an uncut circular plasmid DNA. And notice that instead of producing just one band, there's typically a ladder when you put uncut plasmid DNA, because each of these represents a different level of coiling. So a different number of these supercoils would be present in different versions of this plasmid in the same prep. Even if it's a pure prep of this particular plasmid, each of these represents a different amount of winding. And then over here, if you cut the vector with restriction enzymes, that'll automatically loosen those supercoils and loosen it up into a linear fragment, which would then migrate, as you might expect, this linear piece here and this linear piece here. So this is just a little bit of history. In this particular case, you're looking at three really important studies to reveal that DNA is the genetic material, which again was not readily obvious. In the Griffith experiment in 1928, they observed a change in non-virulent organisms to virulent ones, and specifically they were using Streptococcus species. So a non-encapsulated Streptococcus, which was not virulent, and an encapsulated Streptococcus, which was. And so here's our strain of a colony. A smooth strain is one that is a Streptococcus species with a capsule. So when you inject these into a mouse, the immune response is ineffective, and that results in death to the animal. Here, if you inject a rough version of these bacteria, again, these have no capsule, so they have all the rough contours of the cell surface. Here, these guys are smooth because they've got this smooth polysaccharide around them. So they often form more mucoid or glistening colonies on plates as well. But you inject these guys in, their immune system attacks them readily and kills them. Again, the capsule is anti-phagocytic, and so the immune response has a difficult time clearing them. And when they have a capsule, and therefore the mice are more likely to die. Whereas if you don't have a capsule, the mice are less likely to die. So in this experiment, they heat killed the S strain, which that you would expect nothing to happen, and indeed that's what happens. These are dead, you put them in, and they survive. But in the expansion of Griffith's experiment here, you put the heat-killed S strain in with live R strain. Now, technically, this is what should happen, right? But Griffith showed that the mice basically died, and that's where he came up with this concept that something about these dead microbes transformed these live ones, these live R ones, into smooth live strains that killed the mice. But still, they didn't know what that transforming principle was. So these this is just a paraphrasing of this, which is actually the picture version of this text, so it might be easier to follow. So in the in the experiment by MacLeod and McCarty, pictured here, they knew what macromolecules were present, so they performed this sort of subtractive assay to get rid of each macromolecule individually. So the question is, if I take the R cells and mix them with type S DNA extract, and basically type S cells, you just kind of kill them with heat and then do a crude extraction, and then you treat them with a protease, which would subtract proteases from the mixture of macromolecules. Mix that in there. Do you get transformation? And still you get transformation. Great. So we know that that loss of protease does not interfere with the transformation process. Same thing here with RNAs. So you got the type S extract, you put RNase in there to selectively destroy RNA molecules, but still we get transformation. So again, we know that RNA is not the essential transforming principle. And then obviously they repeat this with DNA ACE added to selectively remove DNA, and obviously transformation is completely blocked. So we know now that DNA is the transforming principle. And then, of course, Hershey and Chase back this up with their experiment using bacteriophages, which, again, I always jokingly say it's fortunate that they chose a bacteriophage that has a DNA genome, or a virus in general with a DNA genome, so this could actually work. Imagine if they chose a RNA virus to do this work. It would totally be confusing. So again, in this case, sulfur labeled proteins that make up the protein structure of this virus, have them attached to cells of E. coli, and let them go through the initial phases of the infection process, and then you shake them off, use a blender, and then basically track where the radioactive sulfur ends up. This is a S35, and so the S35 stays with the capsid proteins and tail and tail fiber proteins. So no S35 ends up in here. So that can't be, the protein can't be the genetic material. And conversely, when you do the same type of experiment, but with radioactive P32 labeled DNA in the phages can infect, inject that material, and then obviously all the P32 ends up in the progeny of those E. coli. So again, that supports the idea that DNA is the genetic material.